And our next speaker is again on protein nucleic acid interactions is Yaron Orenstein from, again, MIT.
But this time it's to RNA and not DNA. That's the unique part. And I'm going to describe our algorithm to infer RNA binding preferences from RNA-Compete data. So I'll start with the motivation slide. That we know that protein-RNA interactions play roles in many processes in the cell, including the capping of an mRNA, the splicing in different forms, an export from the nucleus to the cytoplasm, an effect on the stability as well as the translation efficiency. And these interactions between the protein RNA depend both on the RNA sequence as well as the RNA structure. So when we are saying RNA sequence, we all know that we are referring to a string over the nucleotides ACGU. But what do we mean when we say RNA structure? So in this study and in many other studies, when we are saying RNA structure, we are referring to the RNA secondary structure. And this structure is defined by base pairing of nucleotides, where the canonical nucleotides would be A and U, and C and G, and we can also have some non-canonical base pairs such as U and G. So at the end, we can take an RNA molecule and efficiently predict, using some software such as algorithms from the Viano package, the structure. But in many cases, it's not that the specific base pairing is important for the protein to bind the RNA, but it's the structural context that different nucleotides have. So for example, we may see this stretch of nucleotides being in the context of an hairpin loop, which is induced by the RNA structure. So now we are interested in the context of the different nucleotides, and in this study we are going to consider 5 contexts: the paired hairpin loop, multi-loop, inner loop, and the external contexts, which is not encompassed in any stem. Now, RNAs have one minimum folding energy structure, but in fact they can fold in many different structures, and the number of structures they can fold into is an exponential number. So we cannot take into account all possible structures, but there are several solutions of how to deal with this exponential number of structures. So one way is to look at representative structures. So we can run an algorithm called RNA shapes, which give us a set of structures which are representative, meaning that if we have 2 structures which only differ by 1 base pair, they won't be considered different structures. But if they have an additional hairpin loop, then they would be different structure. And we can set some energy threshold and say to the algorithm, give us all structures, graph-like structures that are below some energy threshold. Another way to represent the ensemble of possible structures is using aggregated probabilities. So here we have a probability, a distribution for each nucleotide in the sequence to be in each of the structural contexts. So we get a distribution, for example, over the nucleotide A of being an external, bulge loop, multi-loop, hairpin loop, or paired loop. And this can be predicting using RNA-PL fold from the Vienna package, or experimentally measured using protocols such as IC shape, which gives us the probability of a nucleotide being paired. So how do we measure protein RNA binding? We can do this in vivo using the CLIP protocol, where we start by cross-linking the protein to the RNA. That goes through some digestion process, site selection, pulling off the bound RNA, breaking the protein, and sequencing the bound RNA molecules. But what are the disadvantages and difficulties in this protocol? So first, when we look at the mRNA in vivo, it is usually a long molecule, and that means that it's hard to predict its structure. As well as there are many confounding factors that affect the structure of the RNA. So even when we do the prediction, we don't get an accurate prediction of the structure. Second, we may have cofactors that bind the protein together as a complex, or competing proteins that affect the binding of the protein to a specific molecule. We may have many technological biases in the processes of this protocol, and we also need to measure the background sequence to know what it was the expression of the mRNA or RNA that was available for binding. So to avoid all these complications, in vitro protocols were developed. And here I'm referring to the protocol of RNAcompete from the lab of Tim Hughes from University of Toronto. In this technology, we start with a set of 240,000 RNA probes, all designed to be unstructured, and they have a mathematical property that together all RNA sequences cover all 9 mers, where each 9 mer appears at least 16 times. The protein can bind this spool, then the bound RNAs are pulled down and led to hybridize to a microarray that has the complement sequences. So by the intensity on the microarray, we can figure out how much protein was bound to each RNA molecule, and the final output of the protocol that computational people like me look at is a set of sequences and the binding intensity that corresponds to each of these sequences. And here are the advantages compared to the in vivo protocol, that first, all of these RNA sequences are between 30 to 40 base pairs. So the prediction of their structure is much more accurate, as well it is in vitro, so it doesn't have any confounding factors. We only test a single protein, so there are no competing proteins or cofactors in the assay. There are less technological artifacts. This is an in vitro protocol with less steps, and we can also measure a background signal on the microarray before we start the experiment. So what is the computational challenge that we solve in this study? So 3 years ago, a study from Tim Yu's lab by Ray et al. Published 244 RNA experiments on unstructured RNA probes. And this was, and still to date, the largest set of in vitro experiments done measuring protein RNA binding. Unfortunately, in the same study and in following study that looked at the data, only sequence preference were derived from this data, by making the assumption that since the RNA probes were designed to be unstructured, we cannot derive any structural preferences of this protein. So the challenge that we take on in this study is to infer both the sequence and structure-based binding preferences from this data. What are the extant solutions to solve this problem? So together with the RNAcompete technology, an algorithm was developed from Quaid Morris lab, also from University of Toronto, called RNAcontext. In this model, we model the sequence preferences and the structure preference using different parameter sets. The sequence preference is modeled using a position weight matrix, and we have another 5 parameters for the different structural preferences, and the parameters are inferred using the LBFGS optimization procedure. Another algorithm, the state-of-the-art in inferring protein RNA binding preferences, is GraphProt, that was published 2 years ago from the lab of Rolf Backhofen. Here, the features on the RNA molecule are small subgraphs that are in some distance between each other, and additional features are features of the hypergraph, which gives us the structural context. And the weight of these features are learned using SVM or SVR, depending if it's a classification or regression problem. What are the limitations of these methods? So first, for RNA context, it is that the fact that the sequence preference is modeled by a position weight matrix. And we all know that an inherent property of this model is the position independence. So if we have dependence between nucleotides in the binding site, we cannot model them using this model. In addition, there is an assumption that there is a global structure preference, meaning for any nucleotide sequence, we have the same structure preference. And this may not be a valid assumption. For the GraphProt case, the limitation when applied to the specific unstructured data of RNAcompete, that the structure is represented by graphs, and when we try to predict structure as graph on these unstructured probes, we'll just get a linear sequence. In addition, the runtime and memory is quadratic in the data, so just running the algorithm on one RNAcompete experiment takes more than 7 days. So we need a new algorithm to solve the problem for the RNAcompete data. So here is the model that we developed and our contribution in the study. So we call it RNAcontext-kmer, since we extended and taken the original RNA context and expanded it into a kmer space. First, we take the position weight matrix, which model the sequence preferences, and instead of having just a PWM, now we are going to have a sequence score for each different kmer. For the structure preferences, instead of having global structure preferences, now we are going to have different structure preferences for each of the kmers. So going from a linear number of parameters, now we are in an exponential number of parameters. We again use the LBFGS algorithm to perform optimization and learn the parameters, but we needed to improve the implementation because in the original implementation of the RNA context algorithm, the running time of each iteration was the size of the data times the number of parameters. Since we have gone from the number of parameters being linear to exponential, we needed to improve that, and the improved implementation, the running time is the size of the data plus the number of parameters. So now we can run the algorithm in feasible times, and we can optimize the value of k, the width of the model, by doing a cross-validation on the training data, but we found that for most proteins and most experiments, the value is 5. So this leads us to conclude that 5 is the limit of the sample size that we get from the experiment and not a real biological info about the protein binding preferences. Now, a little bit about the model, and this is going to be the only slide with mathematical equations. So we are going to assign a binding score for each kmer W with probability vector P by the sequence score times the structure score, where the sequence score is going to be the logistic function over the kmer score plus some bias term that we learned from the data, where the logistic function scales everything between 0 to 1. On the structure side, we are going to take the average of a specific structural context, the average probabilities over the kmer, and multiply it by the parameter. Again, we fit it into the logistic function. And the way that we learn the parameters is by minimizing the sum of squared errors between the predicted probe intensities and the measured probe intensities, which is found here. And we have a regularization term to keep most of the sequence scores small, and having only a few kmers with high scores. So now to the tests and validation. So first we wanted to gauge how much benefit we get from each one of the modifications that we performed on the model. So we used the original RNAcompete data that included only 9 proteins, and that data also included a few structured probe sequences. And here we compared the move from a PWM model to the kmer model in the sequence scores. We see that for all 9 proteins we improve, or that we do as well. So maybe 3 proteins sit on the diagonal, but all the other improve. When we examine the protein that benefits most from this change, and we look at the top kmers in the model, we see that there is some similarity, but it's not striking similarity, meaning that it will be difficult for a PWM to model the sequence preference, so that's why we get a better performance using the kmer model. Now moving to the second change that we have done, moving from global structure preference to specific structure, which is specific per kmer, we see again a significant performance for almost all proteins, and there is one protein which benefits most from this change. And when we examine the preference for the different kmers being in an unpaired context and being in a paired context, we see that there is one kmer that the protein really likes to see in an unpaired context. So there is no way it's going to bind it when it's paired. This was also found in the original study by aligning the top bound sites, but here our algorithm learned it automatically. Now, moving to some more systematic tests, comparing our algorithm to the state of the art and to extant methods, we went to the data set of the 244 unstructured RNA-compete experiment. What was done there was very elegant, that each experiment was divided into 2 halves, so that we can do the training on one half and do the testing on the other without requiring the cross-validation. And here we measure the performance by Pearson correlation of predicted and measured intensities. We compare our algorithm, RCK, to the original RNA context, deep bind, which is the state of the art in sequence, specificity of protein and DNA and protein RNA binding, and 2 other methods for which the results were published in the deep bind study. We see that our algorithm performs significantly better than the other algorithms in this task of predicting in vitro binding. When we compare in a pairwise comparison to the 2 runner-ups, RNAcontext and deep bind, we see again the significant improvement, but this time in a pairwise plot. When we compared RNAcontext to deep bind, we saw that one was not significantly better than the other. We moved on to test our algorithm and other methods in predicting in vivo binding, so meaning learning the binding preferences from the RNAcompete experiment, which is in vitro, and then testing how well it predicts in vivo binding. For this aim, we used the CLIP datasets that were published in the GraphProt study, and the overlap that we had between the RNAcompete dataset and the CLIP datasets from GraphProt was only 10 proteins. And here are the results. So again, we see that our algorithm, RCK, performs better than deep bind and RNAcontext, but it does not perform better significantly since we are only looking at 10 proteins. And when we look at the different proteins, we don't see one algorithm that performs better in the majority of the proteins. So in this case, we still cannot conclude which algorithm performs better for this task, and we are looking for, hopefully, more data and more overlap between CLIP experiments and the RNA-compete experiments. One of the benefits of our algorithm is the fact that instead of using the predicted structure probabilities, we can use experimentally measured probabilities. So we went to the IC-shape data from our Chang's lab from Stanford, which is the only study that had both the IC-shape data and the CLIP data on the same cells, the same experiment done in the same lab. So we can test how much benefit we get from using experimentally measured structure as opposed to using predicted probabilities. The only protein that was in the overlap between the RNA-compete study and the IC-shape data was the HUR protein, which likes to bind the U-stretch in an unpaired context. It's found by our algorithm as well as by alignment of top-bound bind sites in the RNA context study. And here are the results comparing different structure sources. So here in vivo and in vitro refer to the IC-shape data. In silico refers to running RNA-PL fold to get predicted structure probabilities. None refers to using no structure scores, just using the sequence scores. And uniform is predicting a probability of half for each nucleotide being paired or unpaired. And we see for 4 of the RNA-compete experiments a great gain just by using the structural information, confirming our model that the structure plays a role in the binding of HUR protein. And we see a slight benefit in using the experimentally measured structure, but still we cannot say if it is significant since these are only 4 examples. But this is another motivation for using our algorithm, that now we can use the experimentally measured structure and maybe by that improve the prediction of in vivo binding. So to summarize my talk, we developed a new kmer-based model for sequence and structure RNA binding preferences, and we implemented an efficient algorithm to learn the parameters for this model from RNA-compete data. We showed that our algorithm significantly improves in vitro binding prediction compared to extant methods based on 244 RNA-compete experiments. For the in vivo binding prediction, the task is still open and we don't know which algorithm performs best. And we are looking for more experiments and improvement in their quality. But we hope that through the experimentally measured structure, we might be able to improve the prediction. And through the algorithm, we provide to the community the first of its scale data sets of protein-RNA structure-based binding model, which are publicly available together with the algorithm on this website. So I want to acknowledge and thank Professor Bonnie Berger, my postdoc advisor, and my colleague Yuan Wang that helped in the validation on the IC-shape data, the funding from NIH, and the generous travel fellowship that was provided by HITSEC. And thanks for your attention.